Hello, I'm Michael Dustin from the University of Oxford and New York University School of Medicine. Welcome to the talk on Immunological Synapse, Part 2, Signaling and Function. So this is a movie of free immunological synapses forming, and you can see this bullseye-like pattern emerging from a initial less organized or even inverted pattern. So this is the process of immunological synapse formation. We'll define this a little bit more generally, and then I will go on to talk about signaling, effector function mediated by the immunological synapse, signal amplification at the immunological synapse, and applications to autoimmunity and cancer therapy. So in terms of definition, the concept of a synaptic basis of T-cell activation really went back to the 70s and early 1980s. And there's a review by Mike Norcross in 1984 that describes a synaptic basis of T-cell activation. Bill Paul and Bob Cedar used the term in 1992 in a Cell Review. But the first appearance in peer-reviewed publications is in the late 1990s with work from Avi Kupfer describing supramolecular activation complexes, and our work describing, in reference to Kupfer and our own work, referring to immunological synapses, and that final bullseye pattern is a mature immunological synapse. So what we wrote was that an immunological synapse is characterized by a specific pattern of molecules in the contact. LFA1 is localized in the periphery of the contact, what Kupfer described as the PSMAC, or peripheral supramolecular activation complex, while the T-cell receptor is located to the center of the contact, or what Kupfer defined as a central supramolecular activation complex. This is described in these references from '98. And later on, in collaboration with David Coleman, who's a neuroscientist, we basically thought about a few criteria that were common to the neural and immunological synapses. First, that cells remain individuals, which is part of the neuronal doctrine. That there are bona fide adhesion molecules mediating these junctions, basic cadherins in the neural synapse and integrins in the immunological synapse, that these are at least provisionally stable junctions. Obviously, in the nervous system, synapses can be stable for years. In the immune system, the cells are extremely motile in the steady state, so when they stop for a few hours, that's quite a contrast in their behavior and a relatively stable junction, although it's much less stable than what you'd see in the nervous system in many cases. And you have directed secretion taking place across this interface, and I'll talk about that a little bit when we describe effector functions. So in terms of signaling mechanisms or the actual triggering process, the T-cell receptor, which is again at the heart of the immunological synapse, provides the immunological specificity, utilizes what's referred to as non-receptor tyrosine kinases. So it utilizes several different kinases, but the first two are the Sark family kinase LCK and the Syk family kinase ZAP70, or zeta-associated protein 70, as major signal initiators, which are recruited to phosphorylated cytoplasmic domains of this multi-subunit complex. This signaling process is modulated by a number of other proteins, and this paper published a couple years ago from James and Vale reconstituted this process in non-lymphocytes using a combination of the T-cell receptor, the kinases I just described, and then the modulators CSK, a kinase that actually inhibits LCK by phosphorylating it, CD45, a phosphatase that antagonizes the CSK-mediated inhibition, and together this system would basically set up a regulated basal state from which T-cell receptor engagement would trigger signaling. So this seemed to be a minimal set of molecules required for reconstituting T-cell receptor signaling in a non-T-cell. But when we go back to T cells and basically look at these molecules, we can see that they're very highly organized in the immunological synapse. So a number of the experiments I'm going to describe utilize a reconstituted system in which the antigen-presenting cell is replaced by a supported planar bilayer, which is kind of schematized here, which, again, a technology developed in Hard McConnell's lab at Stanford, where artificial bilayer is deposited on a glass substrate. This is a very optically ideal system. We can put purified molecules in a laterally mobile form in the substrate, and T cells can assemble an immunological synapse. We can then use optical methods that are only applicable to this type of interface, like a total internal reflection fluorescence microscopy, in which you're bouncing a laser beam off of the interface and creating this very shallow evanescent wave that excites fluorescence within around 200 nanometers of that substrate. So it's very synapse specific in this case. And for example, we can then look at signaling processes like the recruitment of ZAP70 to these T-cell receptor clusters, which are actually the active component in the signaling immunological synapse. So you have this large central cluster, which is actually not active in signaling, but these small peripheral clusters are the places where most of the ZAP70 is being recruited, and most of your other evidence that this tyrosine kinase cascade is localized. We can also see that these microclusters, based on this work from Rajat Verma, which was done in my lab, where if you look at CD45, the signal here, in relation to the T-cell receptor on these planar substrates with a MHC peptide complex that triggers this T-cell, that you very early on in the contact or later, once you have this mature bullseye-like immunological synapse, that you have CD45 exclusion from these microclusters. And this is where the ZAP70 is being recruited. So while one hypothesis for triggering through the T-cell receptor is that the local exclusion of these phosphatases like CD45 would be critical for this. And this is an idea that was first floated by Tim Springer and has been expanded on extensively by Simon Davis and Anton van der Merwe, and certainly supported experimentally, at least as a kind of in a descriptive fashion here. So another remarkable thing about both the immunological synapse and this total internal reflection fluorescence microscopy as a method to study it is that we can detect single MHC peptide complexes, and we can actually basically start to study the sensitivity of the T-cell and directly demonstrate this single molecule recognition process. So these are single MHC peptide complexes that are being captured. They're diffusing around on this planar bilayer substrate, and the ones that are immobile here in this red area, which is actually the T-cell receptor, are effectively being captured by the T-cell receptor. The T-cell has about 50,000 receptors on its surface. Maybe over 10,000 of those are in this interface, and those are there waiting in a proper environment defined by the adhesion molecules to very efficiently capture these molecules. So to point out that the LFA1 ICAM1 adhesion system is in place in this system, it's just not labeled in this context. So you'd only achieve this very high sensitivity with the adhesion system in place, and actually, if we looked at it, it would be forming this ring-like PSMAC. There's not a large CSMAC in this case because the CSMAC size is linearly related to the MHC peptide density, and at very low MHC peptide densities, you don't see much. We'll talk more about that in talk 3. So with this single molecule recognition process, we looked for CD45 exclusion to see if this CD45 movement from those sites was seen even when the T-cell receptor was just seeing a single MHC peptide complex. And what you can see from this plot is that we did these measurements of where the single MHC peptide complex was engaged, and then at the point of engagement, and then around that point, and then basically did a local comparison, set that to 1, and then basically looked at what was there, the surrounding area was set to 1, and then we would look at what was happening in that central region. And what you can see is that there's a lot of noise in these measurements, but that we can still see exclusion of CD45 on the order of 10% from these sites of single MHC peptide engagement. So again, from just kind of a phenomenological standpoint, the CD45 exclusion is happening at points where the T-cell receptor is engaged, even by single molecules. And we know, again, that signaling is being initiated there based on ZAP70 recruitment. So one model for T-cell receptor triggering is this, that accounts for the single molecule sensitivity, is this CD45 phosphatase exclusion model. Again, van der Merwe and Davis would be sources to look for more information on that if you're interested. There's also, there are other ways to do this, though. So Art Weiss has demonstrated recently that CSK inhibition can also trigger T-cell receptor signaling using a pharmacological agent. So either CD45 as the sort of negative regulator of local tyrosine phosphorylation, or CSK to regulate LCK will achieve this rapid activation. Balbino Alarcon and colleagues basically have been studying conformational changes in the T-cell receptor and evidence of that. This is again a ongoing area, and no one has a structure for a complete T-cell receptor with the signal transduction component, so we don't know at this point if this is right, but he has signatures of these conformational changes. Kai Vuckefinig and earlier even Larry Stern have described the kind of a conformation of the cytoplasmic domains attached to membrane lipids, and this modification of the sequestration by lipids in the inner leaflet of the membrane can also be used as a triggering mechanism. And finally, Cheng Zhu has come up with some very interesting data suggesting that the T cell receptor, again, in a kind of a conformation change mode, acts as a catch bond that in response to force undergoes conformational changes that both increase the strength of its interaction with the MHC peptide complex and contribute to the signaling process. So again, there are a number of things going on in this area, and the jury is still out on what combination of those mechanisms or even unknown mechanisms that people haven't thought of are actually acting to account for this high sensitivity. So this centripetal movement, again, of the MHC peptide complex and the T cell receptor and the adhesion molecules is very dramatic. And this movie from Ron Vale's group, generated by Adam Douglas, using the Jurkat cell line expressing fluorescent GFP, GFP actin, beautifully illustrates this centripetal movement using a method of speckle microscopy, which is based on a spinning disk confocal microscopy. And so this is clearly an engine for the formation of these centripetal patterns, and the association of the differential association of the T cell receptor and the integrins with actin probably then account for this bullseye-like patterning. In talk 3, we'll also discuss how the escort machinery may be important for actually the final deposition of T cell receptor in the center. So effector function so there are two kinds of. I've talked about signal integration, and the signal integration can accomplish transcriptional changes and cell cycle regulation that leads to the proliferative burst. But once that proliferative burst is over, the T cells become effector cells, and they have to do things. They actually can't just proliferate in things. They actually have to kill targets or help B cells. And this synapse also helps in this stage. And this is a fantastic movie done by a method called lattice light sheet microscopy, which was developed by Eric Betzig at Genelia Farms, and this Ritter et al. Paper with Jillian Griffiths and Jennifer Lippincott-Schwartz beautifully shows T cells with a green fluorescent actin probe and orange markers of their cytolytic granules, which contain agents that will kill this target cell, and also the centriole, the microtubule organizing center, marked with this little dot here. So now if you watch this movie, this T cell really goes after this target cell, forms an immunological synapse. You can see with the clearing of actin, kind of a bright actin structure at the periphery, and then a clearing of actin towards the center, where those granules and the microtubule organizing center dock right up to that interface and deliver cytolytic agents that will result in, again, the death of the target cell. So this is a beautiful example of effector function. Again, this was published this past year. So in terms of thinking about how important is it that you do that to kill a target, so we were able to do some experiments in collaboration with Yuri Sokolev, where we basically could kind of measure the efficiency of a cytolytic T cell by comparing how many granules it released, which is been measured through a serine esterase release assay, and how efficiently it killed the target. So you could kind of use target killing over granular release as kind of a measure of efficiency, and then set up situations where the T cell either formed an immunological synapse or had a broken immunological synapse. So it could basically, pharmacologically repair or break the synapse depending upon, experimentally. And what we found is that basically forming an intact immunological synapse improved killing about 3-fold. And you could say, well, that's not that much. Some biologists have a cutoff at 5-fold. But we actually think this could be very important in the context of, say, killing in vivo, particularly tumor cells, where it can take up to 6 hours for a T cell to kill a tumor cell based on work from Philippe Busseau. So a 3-fold change in efficiency would take things from 6 hours to 18 hours. And then you're actually starting to deal with time frames that are relevant to the replication rate of the tumor cell. So if you want to keep up, basically, it's probably good to have an intact synapse, and that seems to be the case, as I'll show you in a moment. So the other thing I wanted to talk about is this the actin, which again highlights both this bullseye-like formation, the formation of this junction that the cytotoxic T cell uses to grab onto the target cell. Also amplifies the signal, it turns out, from the T cell receptor downstream of that early tyrosine kinase cascade. And this is work from Sudhu Kumari in my group that kind of used a similar kind of actin probe to that used by Ritter et al. To follow T cell receptor microclusters and then look at the very specifically at the actin, the amount of filamentous actin at those sites. And one way to basically take these kinds of movies, which can be a little bit difficult to deconvolve, and illustrate events that are happening over time is to use what's called a chymogram, which gives you essentially this region here. This line effectively is the top for the T cell receptor and for the actin of this graph. And then you basically just run that same region at different times for both signals. And what you can see is that within this very complicated pattern in the actin, you have very steep diagonals, which mean things are moving very fast. They're doing a lot of movement in a short time. But then these kind of more shallow, these kind of somewhat larger angles going down in this direction that basically reflect slower movement, and those are tracking perfectly with the T cell receptors. So you have these foci of actin that are following the T cell receptor very closely. And what Sud was able to show is that these actin foci were dependent upon a protein called Wiskott-Aldrich syndrome protein, which is deficient in a primary immunodeficiency where you have B cell and T cell dysfunction and also platelet dysfunction. But it's basically a actin regulator, regulates the so-called ARP2/3 complex that makes branched actin networks. And if you don't have WASP, you don't basically form these actin foci at the T cell receptors, but a lot of your other actin polymerization is fine, and you can actually form a synapse just fine. So that was a bit confusing initially, but looking at that again, just that actin at those T cell receptor foci was critical for understanding this. And Suda went on to show basically that phospholipase C gamma recruitment is critically dependent upon these actin foci, and phospholipase C gamma is a critical sort of nexus or focal point for T cell receptor signaling. So activating PLC gamma gives you both calcium and RAS activation. So it's both important for acute signaling and also for some of the sustained signaling that leads to transcription. And this is critically dependent upon these actin foci. So these actin foci are not just forming the whole synapse, or F-actin is not just forming the whole synapse, but there are subtypes of actin in the synapse that are driving this signal amplification process. And this is just a kind of a movie that kind of illustrates, again, the remarkable things that are happening in the synapse with actin. These little dots are basically microcontact printed regions of a T cell receptor ligand, and the system is backfilled with the adhesion molecule ICAM1, so the cell is spreading on the ICAM1, actin is polymerized at the T cell receptor, and then it's forming these intricate kind of spirals which basically with the actin kind of distributing to the integrins, and the integrins kind of taking this actin that's being generated at the T cell receptor and then kind of modifying it. So there's quite a bit to learn about the specifics of this, but at this point, one thing we feel we know is that this is very important for signal amplification. So I wanted to talk a little about the applications of the immunological synapse at this point. So having told you some things about the way it's working, so what is the significance of this? So we think that there are a couple of particular things. I mean, one, recent work with Kai Vuckenfennig's group on autoimmune disease I want to point out, and also a recent collaboration we did with a group of cancer immunologists who use radiation therapy in combination with a new type of biologic called a checkpoint blockade drug that essentially, these checkpoint blockade antibodies have really revolutionized melanoma therapy at this point. So kind of really changed the game in a lot of ways. But first, I want to basically define one other term, or at least mode of interaction between T cells and substrates, and this is essentially compare a synapse and what we refer to as a kinapse. So, and just to illustrate what this is, so we have a synapse on this side, which kind of maybe a movie you've seen before. It's radially symmetric. The T cell receptor goes towards the center. This is showing a T cell receptor signal in this case. But you also see cells that essentially start to form a symmetric synapse, but then they break symmetry and they start to move. And this one actually is moving kind of down and towards me. So what you'll basically start to see is that it starts to have a trailing edge and a leading edge. And we can describe the cytoskeletal dynamics of Kupfer's supramolecular activation complexes as having characteristics like a lamellopodium, a lamella, which is basically a site of adhesion molecule concentration for the PSMAC, and then the CSMAC being sort of like the uropod in this migrating cell. So we think that the machinery is actually quite similar. So when a cell is basically moving or engaged in this, what we refer to as a kinapse, a moving junction, it's actually integrating signal in much the same way as the cell that's forming the radially symmetric synapse. But this cell will basically move away from the place where it started out, whereas this cell will pretty much stay in place because the radial symmetry balances all the forces and the cell will stay. So essentially this is an issue of, should I stay or should I go? The cells do these different behaviors in different contexts in vivo, and we've found that this is potentially useful in both the autoimmune and cancer immunotherapy context. So think about it. And we know genetically, based on a paper of Tasha Sims in 2007, I guess in my, again, postdoc in my lab, that essentially the symmetry breaking is driven by a protein kinase called protein kinase theta, and the reformation of a stable synapse from this state going to a stable synapse requires a WASP, the kinase I described before. And again, it's probably in synergy with the integrin system to basically essentially repair the broken piece MAC and basically get you back to a symmetric state. So this heat map, it's complicated, but the punchline is that basically autoreactive T cells, and these are T cells from patients that recognize bona fide autoantigens in multiple sclerosis and type 1 diabetes, and comparing these to T cells that are responsible for killing flu-infected T cells in influenza, influenza-infected target cells, essentially these form fantastic synapses. So the red and orange all say that these are, attributes of stable synapse formation. And then as you basically go into these different clones, which are indicated up here in the multiple sclerosis or type 1 diabetes setting, you see, progressive, essentially defects in this immunological synapse formation process. So I guess, and this is just kind of illustrated here, you have essentially kinapses forming with these MS-specific T cells and type 1 diabetes associated autoreactive helper T cells. So this is, and again, this is a characteristic that appears to come from the antigen recognition process, self-antigen recognition in this context, driving a weaker synapse. And we think one possibility is this failure of stable synapse formation may make these more difficult to regulate. That's our hypothesis at this point, and I think this is something that we're looking to test in the future. So cancer. So immune evasion has recently been made by cancer biologists, this review by Hanahan and Weinberg that kind of revisited the hallmarks of cancer. So cancer immune evasion is now considered a hallmark of cancer. So this is a big change over the past decade in how people think about this. And a lot of this change has been driven by a couple of advances based on these therapeutic agents that basically remove inhibitors of the immune system or neutralize inhibitory pathways that are probably normally protecting us from autoimmunity or immunopathology, but in the context of cancer can be allowing the cancer to evade the immune system. You block these and the immune system suddenly will see the tumors much more clearly and destroy them. So again, a change in opinion. And this is one of those systems, so an antibody called ipilimumab that's targeting a protein called CTLA4, which is a cytotoxic T lymphocyte antigen 4. And this is an image from work by Jim Allison and Jackson Egan, basically showing a fluorescent CTLA4 moving to the immunological synapse. So this is a synaptic marker. It's a regulator of effector T cells. It's generally thought of as an inhibitor. When it's blocked by antibodies, it then increases activation. Essentially, it also is used by the regulatory T cells I mentioned in part one, which buffer responses, and therefore, because CTLA4 is an effector molecule of the regulators, inhibiting it actually it neutralizes some of the function of the regulatory cells too. And finally, CTLA4 cross-linking, though, has a kind of a side effect. I mean, all drugs have side effects. And in this case, it's synapse destabilization. So again, CTLA4, perhaps because it's involved in an axis that regulates protein kinase theta, which I mentioned before as a synapse breaker, may enhance the activation of PKC theta, and in doing so, essentially destabilizes synapses. And we've actually seen this in a model of cancer therapy in the mouse, where we're setting up a model with a breast carcinoma implanted in the flank of a rodent that then grows over a period of about 12 days and metastasizes to the lung. So it's a model that basically recapitulates key events in the development of breast carcinoma, although it doesn't start from the orthotopic site like in the ducts and the mammary glands. It basically is we're taking the cell line and putting it into the animal. So this tumor is susceptible, and we can then view the T cells within these tumors using a method called 2-photon intravital microscopy, in which an anesthetized animal is basically placed on the microscope stage, kept warm, kept alive, but under anesthesia, and the tumor can be surgically exposed and imaged using pulsed infrared lasers that basically allow you to image deep into the tissue and do time-lapse imaging in real time. And these are some images, and this is basically just illustrating our therapeutic model. So if we put in anti-CTLA-4, again, the mouse, an anti-mouse version of ipilimumab, what we find is that, as in some patients, the therapy fails. So the tumor continues to grow, you have high metastases. The blue cells here are the tumor cells. The red are some tumor vasculature, which again is famously abnormal. But nonetheless, the T cells are getting in. But what you can see the T cells are doing is they're highly motile. Even in the tumor, the T cells are moving. This means they're forming kinapses. They're not forming synapses. However, if we combine the anti-CTLA-4 with radiation therapy, which is the therapeutic modality that Sandra Demaria and Silvia Fermenti were focusing on in this case, now we see that the T cells, the green cells, the T cells within the tumor are arrested on the tumor cells. So they're now forming synapses, and this is correlated with tumor shrinkage and low metastasis. So what we went on to then show, and these are basically, these just illustrate the two modes. So the low motility in the tumor associated with the, again, effective combination therapy, or the high motility associated with the anti-CTLA-4, again, kind of side effect. We found that we could basically recapitulate, even in the combination with radiation therapy, we could recapitulate this increase in motility by inhibiting another receptor called NKG2D. And this is, I don't want to, obviously the details of this aren't important, but this is a kind of what's referred to as a co-stimulatory receptor. So CTLA-4 antagonizes the activity of CD28, which is one type of co-stimulator. NKG2D is in another class of co-stimulators on the CD8 T cells. And what appears to happen is that when you irradiate the tumors, they upregulate ligands for NKG2D, and that additional co-stimulatory pathway stabilizes the synapse. So unlike CD28 that activates PKC theta and inhibits synapse formation, NKG2D is delivering activating signals that are stabilizing the synapse without activating protein kinase C theta, we believe, and this is leading to essentially reversal of the therapeutic effect. So we think that what we're essentially, and this is just illustrated here, so if you block the NKG2D, you kind of increase the lung metastasis versus the effective ionizing radiation 9H10, which is the anti-CTLA-4 antibody. So no lung mets, and basically the antibody to NKG2D, you get the lung mets back. So essentially the behavior of the T cell in the tumor correlates very strongly with our clinical outcome in this rodent model. And this approach is in human trials and is showing promise in conjunction with ipilimumab. So this is sort of an illustration of how we can use information about the immunological synapse to think about what's happening in the context of immunotherapy. And this is a movie of a situation where the combination therapy has been very successful in eradicating the tumor, no tumor cells, lots of T cells. So at least in the immediate post-tumor site period, this is probably what you want to see. So there are a number of factors that will basically be controlling this. But I think one of the key things that we're kind of coming back to is that the radiation therapy is basically triggering an innate injury response. It's upregulating NKG2D ligands on the tumor cells, and this is allowing the immune system to get back on target in the presence of the therapy with anti-CTLA-4 to eradicate or at least significantly slow down the growth of the tumor. So this anti-CTLA-4 side effect of synapse destabilization can be counteracted in this case by radiation therapy. But we think the more specific thing that's doing is engaging this other signaling pathway that stabilizes the synapse, and essentially this what's referred to as a stress-induced molecule, the NKG2D ligand called RAY1, which is another detail. Again, it's in this paper. So again, that's an example of using information about the immunological synapse to think about mechanisms in disease and disease treatment. I'd like to acknowledge many investigators who contributed to this at both particularly NYU and more recently Oxford, where we've moved in the last 2 years. Again, there are many other things you can follow up on in terms of citations in the talk, and I guess happy explorations. So thank you. Bye-bye.